welcome back to your Unit 1 BPCC-102. And in this unit, earlier we were discussing different research methods in biopsychology. So we will be continuing with that. As you can see, the objectives that we have planned for this unit are research methods in biopsychology along with research ethics in biopsychology. So you might be wondering, why are ethics necessary? So this is something that we will discuss further ahead in our understanding of research in biopsychology. And if you remember, in the earlier class, we had discussed one of the methods of biopsychology, but we were not able to discuss all of them. So we will be proceeding with the rest of the methods in today's session. Hope you can see this better now. All right. So as I was just saying, that today we will be discussing different methods to study biopsychology. And in the last class, we had done the ablation method of studying different aspects in biopsychology. So today the research methods that are present in biopsychology, which we will be covering, are histological method, electrical stimulation, psychophysiological recording methods, neuroimaging, and stereotaxic lesion along with chemical stimulation. So these are all the few methods that we will be covering today. All right, now let's come to histological methods in research for biopsychology. Now, you know that primary research in biopsychology is dependent on the fact studying the brain and how brain and nervous system influences different actions that take place in our body. Okay, so this is why a lot of techniques within the field of biopsychology could be invasive in nature. So there are several techniques and methods to study anatomy and functioning of human behavior. But when you want to study very small details of a tissue, or when you want to take place a microscopic investigation, histological methods are the ones which are used. So whenever you have to do a microscopic investigation in the field of biopsychology, you opt for histological methods. Now, what happens in histological methods is that a sample of the brain is under investigation, and hence it is prepared so it can be mounted on a slide and observed under a light microscope. So mounted on a slide means, if you have ever entered a biology lab during your 10th or 11th, 12th, you might have seen that there was a graft sample we used to do for onions, right? So similarly, we used to graft and mount the cells, so that the same thing happens, that you cut a tissue from the brain and you mount it on a slide. So this slide is usually transparent in nature because it needs to be placed on a microscope. Okay, so in order for any tissue to be studied in microscopic condition, this is a necessary aspect. So over here, the neural tissue is extremely fragile and it needs normal condition. So what happens is, if we keep the neural tissue in normal condition, it will disintegrate, meaning it will dissolve or get destructed. Hence, we need to fix the tissue by staining it in order to view it on the slide. So staining is primarily coloring the tissue and putting it in a protective fixture to make sure that this tissue does not get destroyed. All right, and when it is taken out, the bacteria or molds tend to decompose the neural tissue. So this is what happens with the neural tissue, because it's a very fragile tissue of the human body. Okay, now when we talk about the histological methods, there are 4 primary methods here. So we have our staining method, we have fixation, we have freezing, and we have embedding. So when we talk about histological methods, they are divided further into 4 aspects. So I hope you have understood what happens over here. See, in ablation, we focused on studying lesions. However, in histological method, you might create a lesion. However, the study is entirely microscopic in nature, hence the focus is on a very small unit of the brain or the smallest unit of the tissue, because we want to do a microscopic investigation. Okay, moving ahead next. All right, so when we talk about the histological methods, as I discussed earlier, that there are 4 of these methods, and first we will be discussing fixation. So when we talk about fixation as a method, what happens in this method is a fixative solution is used to fix the tissue. Now, what is a fixative solution, you must be wondering. So fixative solutions is often something that would keep the tissue freezed. All right, so it keeps the tissue freeze and embedded for the microscopic observation. Okay, so making sure how to see neural tissues are fragile and we cannot keep them in normal conditions. Hence, fixation is done where you harden that tissue so that you can observe it in microscopic condition. So you put a fixative solution on it which stabilizes the neural tissue. Okay, so over here what happens is first you have to clean the tissue and then you have to dip it in a fixative solution. Now this solution in most of the cases is formalin. However, other fixatives are also used. Okay, but we know now that since this method has been used over the period to study neurons and neural cells in a microscopic light, we know now that this fixative prevents autolysis and makes the tissue hard. Okay, now you must be wondering what is autolysis. So autolysis is the self-digestion or breakdown of cells by their own enzymes. This is why we cannot keep neural cells at the same or normal condition, because they would eat themselves. Okay, and hence we use the method of fixation, histological methods. Next comes freezing. So as the name suggests, in freezing we froze the tissue in order to be sliced. So to freeze the tissue, it is often dipped in a sugar solution and then put in a freezer. So the temperature is regulated in a way so that the tissue is maintained at an appropriate temperature, neither too cold nor too hot. Okay, and once the tissue is hard enough, it is placed on the microtome to be sliced. So you will cut the tissue in a very thin manner so that you can study it further. Okay, next aspects of this comes are embedding. So embedding is another method of assessing or utilizing the histological method. What happens in embedding is, this is one of the ways to make the brain tissue hard by embedding and covering it by paraffin or nitrocellulose. You know what paraffin is? It's a type of wax. So often this is used to harden the brain tissue. And once the tissue is hard, then we cut it using a microtome. So microtome is a slicing device which is used. Okay, so these slices are further placed on the slide and mounted on it using albumin, which is from the egg. So albumin is something which is present in the egg and that is used for mounting and fixing the whole tissue. And then once the slide is dried, it is dyed using different chemical solutions. So see, you can notice that most of the methods in histological method require you freezing the tissue and placing it on the microscope to study. So this is why it's not that invasive in nature. However, it still requires you to freeze the tissue, cut the tissue, make sure you make a proper mounted slide out of it so you can study. So all these aspects require a lot of surgical elements and skills. Okay, coming to the last method in histological method, which is called as staining. So staining is done so that the structural detail of the tissue becomes apparent. So how do we know which structure belongs to what? So let's say if you have a tissue in your hand and it's not stained, okay, let's say it's a brain tissue and it's not stained, we will not know which part is where. Right, because that's a marking we cannot do. So hence staining is used, and it is done when we darken or color particular features of the tissue. So often we use Nissl stains or cell body stains to dye the cell bodies, and myelin stains are often used to dye the myelin sheath. Okay, so these myelin sheath are the nerve pathways. So through myelin stains, we can study the nerve pathways. Okay, and then we also use membrane stains like Golgi-Cox stain to help us identify and color the cell body, dendrites, and axon. So through this, we are able to understand the functioning of these small bits of the brain cells as well as the tissue through staining. Without staining, we won't know what action is happening when. Okay, so these were the 4 techniques that are utilized in histological method. I hope which is clear to all. All right, now let's move ahead to the next aspect, which is the psychophysiological recording methods. So when we talk about the psychophysiological recording methods, these are different. So there are a variety of these. So we have electroculography, we have electrodermal activity, we have electromyography, magnetoencephalography, and electroencephalography. So this is known as EEG, MEG, all right, EDA. So these are all the short forms and they are the non-invasive way. Now, ablation and histological methods both are invasive. Why? Because it requires you to cut certain parts of the brain or to cut or take a tissue from the brain. So all these things are necessary in those techniques. However, psychophysiological recording methods do not require any invasion. And through this, you can study the brain and engage in biopsychological research from outside the skull as well as other parts of the body. Okay, let's discuss these ahead. So, as you can see, the first one is that electroencephalography, or we call it as EEG, something that if you go for internship in a psych institute such as NIMHINS or any other place where they do psychological assessments, you'll find these machines. In general, you might hear people say that, the EEG was done to understand the brain function. So it's a common fact of research within the field of biopsychology. So we know that brain works under the electrical circumference, right? So there are certain electrical activities of the brain which can be recorded through the EEG machine, and this technique is known as electroencephalography. So what happens in EEG, as you can see that the individual skull is covered with certain electrodes, and you can see in the background there is a paper. So that's the EEG reading that is coming, which is showing the electrical activity. And you can see on the skull there are different electrodes, right? So electrodes are placed on the scalp of the client or the subject or the participant, and recording is done through the computer, which you can see in the background. So earlier, when we did not have so much technological advancements, what used to happen was EEG signals were often detected or recorded on the rolling paper, okay, where the reading were displayed. But now we can take a computer printout, and these signals are pretty long. All right, they're like very long signals, so it takes a lot of time to read them. So EEG signals often help in the diagnosis of various disorders of the brain: mild head injury, cerebral pathology, epilepsy, and speed of processing information. It also helps us in understanding how the memory functions affect or get influence with an increase in age. So this is a common tool which is used in memory research as well as by several clinicians who are trying to understand the pathological illness or pathology of their clients. All right, moving ahead, next we have MEG. So when we talk about MEG or magnetoencephalography, this requires magnetic fields. Earlier, that was electrical fields and electrical activity, but MEG requires magnetic field. Over here, what happens is MEG measures the change in magnetic fields on the surface of the scalp by producing changes in the basic pattern of the neural activity. So what happens is, so whatever our basic pattern of neural activity is, that is changed. Okay, so there would be changes that would come. If you have had the chance to study physics, an interesting thing that is that all of us are surrounded by magnetic fields. Okay, so Earth in general has its own magnetic field, which affects the magnetic fields of several other components. So over here, these magnetic fields are often like this. So they interconnect with one another like this. Okay, but what happens in some cases, we can cause changes in the magnetic field by presenting an opposite magnetic field. So that is a similar approach which is followed in this particular method of recording brain activity, is by forming changes or creating changes in the basic neural activity and then understanding the brain functioning. All right, so that is what MEG is all about. Next comes our electromyography. So as the name suggests, it requires electrical stimulation along with our skin Okay, so we will be discussing what happens. You can see the diagram of how usually things happen. So you can see that there is the hand of an individual. We have a stimulator transformer, an input box, active electrodes in the muscle, and we have reference electrode over the muscle along with a stimulator which is over the nerve. So what happens in electromyography is that this records the muscle tension with the help of electromyogram. So EMG, this whole device is known as EMG. Now what happens is the electrodes are attached to the skin over the muscle which needs to be investigated. So let's say if I have to investigate this aspect of my hand, I will be attaching electrodes over here. Okay. So when the muscle contracts, producing some amount of muscle tension, the tension would be recorded, and this information often helps us in understanding muscle tone. So this is a great method of research which can be used and is still in discussion of use for individuals who often suffer with muscle dystrophy or pathologies that affect the muscle functioning. So this will allow, let's say if I want to understand my hands muscle aspect, so I might insert that electrode over here and then try to understand using the EMG. Okay. Moving ahead, next we have electroculography. So when we talk about electroculography, this is pretty similar in terms of eye tracking devices. So over here, the eye movements are recorded through the use of an electroculograms, which is also called as EOG. So over here, what happens is there is a slight difference in the electrical potential in the back and front portion of the eyeball, and hence this difference in potential is recorded with help of electrodes that are placed near the eye. So if I want to understand this particular difference or how the eye functioning is happening or how eye movements are happening, I would be utilizing this particular tool because it helps me in understanding the difference, and through that I can understand which gaze the person was looking at. Was it a left gaze or a right gaze? So because of that, we often opt for EOG in research. Now there have been some advancements in research which has also led us to different elements of I would say EOG, or this is also neuro tracking or an eye tracking device which helps in tracing where the person was looking when a specific stimulus was being shown. Okay. Moving ahead, next we have electrodermal activity. So electrodermal activity is entirely based on engaging in terms or recording how the skin reactions are. Okay, so over here what happens is there are two main indices of electrodermal activity: the skin conductance level, which is the SCL, and the skin conductance response, which is the SCR. So what happens in electrodermal activity of the skin? So over here the activity of the skin is recorded by attaching electrodes on the fingers and recording fluctuations in skin conductance. This means how well the skin is able to conduct a certain electrical stimulation. So SCL over here measures the background level of skin conductance associated with a specific situation, and SCR measures the changes in skin conductance associated with distinct experiences. So SCL measures the situation, whereas SCR measures the experiences. So whenever there are different kinds of emotional stimuli, there will be a variation which will be produced by skin conductance level due to changes in the sweat gland activity. So over here the entire basis, and this is I would say a technology which is also used in your lie detector machines, where you try to understand the change in heartbeat, the slight change in sweat response. So all these elements come in that. Okay, so I hope the electrodermal activity is clear to all. All right, now move ahead to the next aspect, which is electrical stimulation. So I hope you can see the flow chart again. So when we talk about electrical stimulation, see, up until now we were studying different methods through which electrodes are being utilized. But when we talk about electrical stimulation, this entirely serves and functions on the idea that the brain can be stimulated electrically to understand how it works. So the electrical stimulation system works on the focus that you can stimulate the brain electrically to gain information about the working of its neural systems. Okay, so whenever the brain is stimulated, there is always a change in the resting membrane potential and an action potential is initiated. So resting membrane potential is the potential when your neurons are at rest, whereas the action potential is when your neurons are in movement or they are functioning, okay, or they are acting, right? So that is what happens in action potential. So this causes behavioral changes depending upon which part of the area of the brain has been stimulated. So for stimulating the brain electrically, we often use electrodes, microelectrodes. Okay, so these microelectrodes are made of small metal wires through which the nerves are electrically stimulated. So you will be stimulating your nerves electrically through microelectrodes. And then we also have macroelectrodes, and these are made up of steel wires, and they are often placed either on the top of your scalp or inside the brain to record the functioning of tracts. So these are large number of neurons. Okay, so whenever you want to study how a large number of neurons function together, microelectrodes are used. Okay, moving ahead next, see, electrical recordings, we can make them over a longer period of time. So let's say when an individual is recovering from a surgery and going into physical therapy, that is where we can use. So this kind of, it's often used on animals, and this is known as chronic recording. So if a scientist is focusing to study a particular nerve pathway or where the information is going, then acute recording is done. However, when you want to study it for a longer period of time, chronic recording is done. And these recordings of the electrical signals are often displayed through an oscilloscope or ink writing oscilloscope. Okay, so we will be studying and discussing these three further ahead, how they help us in studying the electrical stimulation. So as you can see, when I talk about the oscilloscope, it helps to display the electrical recording per unit time. So this is a very simple way, or I could say for the scientists who are studying, for us it would be difficult, yes, but what it does, it helps to understand the electrical recordings per unit time. So it has a cathode ray tube on which the display is done. So cathode ray tube is a technical tool over where you can see the recording which is happening, and this recording, so cathode rays are often used to record the activity of a single neuron. So whenever the action is detected, it is amplified and heard by the researcher. So on the tube, this display would come, and whenever the neuron would come into action phase, it would amplify it. The oscilloscope would amplify it, and then we can hear it. Okay, next comes our ink writing oscilloscope. So when I talk about the ink writing oscilloscope, this is also known as a polygraph. Now this evokes so whatever evoked potentials are recorded with the help of various electrodes that are attached to the scalp. So over here what happens is there will be several needles, pen tips, which would continuously record the neural activity on a rolling paper. So this is something that was used in earlier times. So in EEG, what we discussed, right? That the recordings would come in the rolling paper. So that is what similarly ink writing oscilloscope talks about. Okay, so it would be like a pen, and you might see it in old movies when a patient is lying or they're doing a brain test on him. The EEG would be run and it would come on a paper, on the ink, right? So it's more like a fax machine, we can say, for the brain activity. But yeah, that's what it works like. So this helps to detect any tumor in the brain, epilepsy, or you can also study the sleep and arousal patterns of individual. Now modern techniques are used, so you can directly print these recordings, but earlier ink writing oscilloscopes were preferred by several researchers. All right, now computer. So computer is a well-known device, right? It's a technological tool of our generation, and this helps to display, store, and analyze information which is collected by the electrode signal. See how efficient a computer is. I would give you an example, is that this can store up to n number of EEG data, which you can easily analyze in one sitting. Earlier, when it was done on paper, you would have lines and lines of paper, and you would have to sit. If you were a researcher doing research back then in biopsychology, you would have to sit through each of those papers, see the notations, right? But in computer, it's pretty more efficient because there's more, I would say, movement in the data, as well as you can notice that, okay, these were the changes. Identification is easier, as well as you can go through it in one go. All right, so that's the electrical stimulation bit. Okay. Now coming to the next aspect, which is chemical stimulation. Now we spoke about electrical stimulation, which serves the purpose of understanding through electrodes, micro and macro. Now we come to chemical stimulation. So there are several ways how similarly we can electrically stimulate the brain. We can also chemically stimulate the brain and make lesions. So drugs can be given to individuals. So there are several methods. All right. So we have drugs, we have cerebral dialysis. All right. We have immunocytochemistry, and then we have micro inotropesis. So we will discuss each one of these. So what happens often is there are different methods used by different researchers depending on what they want to focus on. So this is something that you have to remember. So when we talk about drugs or medicines in general, right? So what happens is that drugs are given through injections or by inserting a tube into the stomach directly or into a vein or fatty tissue in the body. Okay, so the chemical activity of the brain can be assessed through this. Now, one is by using a 2DG. So 2DG is known as 2-deoxyglucose technique. Okay, so 2DG is a radioactive drug, and when this drug is injected in the animal, researcher waits till it is absorbed by the brain. Remember, anything that goes inside us gets absorbed by the entire body. All right, so researchers would wait it out till it gets absorbed by the brain, and after that, animal is often killed and the brain is removed and slices are made. So see, if someone plans to do these kind of studies or research, a great deal of ethical element has to go in it, because they have to get it approved by the ethical committee. They have to make sure that yes, something like this is necessary, because even when we talk about the lives of animal, every researcher wants to make sure that no life is wasted. So hence they go through lengths in terms of determining whether they really need to use a living individual or not. All right, so this is something that is done prior you move ahead in the research. Okay, so once the animal is killed and the brain is removed, autoradiography is often conducted on those slices, and the slices are then again coated into photographic emulsion. So these are then developed like photo films. So if you have had your Polaroids, so exactly how it is. So they are developed as films, and neurons that have taken the drug appear black spots. Okay, so any neuron which has taken the drug would appear black, or there will be dots on the slides. Okay, now another useful technique that comes in chemical stimulation is your cerebral dialysis. So when we talk about cerebral dialysis, we are primarily talking about a different method of study. Over here, what happens is you insert a tube into the animal's brain directly, not the stomach, the brain. And then the animal is engaged in an activity, and neurochemicals released while the animal is active are collected and then analyzed using the chromatograph. Okay, chromatograph is a color graph technique which is used in cerebral dialysis. All right, so in vivo voltammetry is also one of the techniques which is used to record the neurochemicals released in the animal when they are actively participating in an action or in some activity. Okay, so immunocytochemistry is one of the process which helps to locate the presence of neuron proteins in the brain. So this is also something which is done, and this is something that we know, that each neurotransmitter responds to specific receptors during a synapse. Hence, microintoprosis is a process which involves ejection of charged molecules used to apply drugs or chemicals to nerve cells. And this further helps in locating the specific receptors to which neurotransmitters respond. So what you do in microintoprosis is that you would eject charged molecules through the use of drugs or chemicals to the nerve cells, and then this would be utilized, this knowledge is further utilized to locate the specific cell receptors of the neurotransmitters. Okay, because see, whenever a specific neuron or whenever a neurotransmitter comes into the picture, it would respond, the neuron would respond to that receptor, right? Because it wants to connect, and hence this information is utilized to understand the different neurons and different neurotransmitters. This is something that we will be discussing ahead in detail when we do the neuron chapter and the neurotransmitter chapter. So you will get a better picture of it then. Okay, let's move ahead to stereotaxic lesion. So when we talk about stereotaxic lesion, this is again a invasive technique. Okay, so in this what happens is that in cases where ablation is not possible for small structures that are often located beneath the surface of the brain. Okay, so whenever you want to study something which is beneath the brain or the structure is really small, let's say any part of your hind brain, okay, any part of your hind brain would often, for that you would use stereotaxic lesions. So over here what happens is you have a stereotaxic apparatus, and this stereotaxic techniques is used to damage a structure in the interior or the most inside part of the brain. So what happens is that first it is important for the researcher to identify which part of the brain should be investigated. Once this is investigated, the stereotaxic atlas helps in locating the exact point of the brain wherever we need to do the surgery. So stereotaxic atlas is something that you can see over here, right? This is the whole stereotaxic tool. So this atlas helps us in pointing out the specific area where we need to do the surgery. Okay, once we have pointed out, the head would be held by the apparatus, and the electrode holder is used to insert the electrode or a cannula into the brain. Cannula is a very thin wire or an injection, like I would say it's a very thin object. Okay, if you have seen the needle, that is what ideally a cannula is like. Okay, so then the electrode is implanted permanently, and whenever the animal comes out of the effect of anesthesia, we have tried to observe the behavior. Okay, so over here there's no specific lesion which is being caused, but what you try to do is you try to insert the electrode so you can understand the behavior is still being manipulated. Mind this, that the part of that brain, that interior section of the brain, is being damaged in one way or the other. All right, so once the study is over, the animal is again given anesthesia to ensure that electrode is removed, and animals often recover from it. Why do you think it is important for animals to be put under anesthesia while we are doing such experiments? Yes, because of pain and ethics. So as I mentioned earlier, that every researcher tries to make sure that they are not unnecessarily harming a life or taking a life, and they're making sure that whatever animal they are using, right, it is being respected, as well as they are not being subjected to unnecessary or undue pain. Okay, hence we try to use as much of a precaution as we can as researchers to make sure that we are not affecting the individual. All right, okay, let's move ahead to neuroimaging. So as you can see that neuroimaging is a brain imaging technique. Over here you try to find structural images of the brain. So consider this as photography. Okay, so over here you try to discover structural images of the brain using different techniques. All right, so you can see there are techniques such as X-ray, CT scan, MRI, PET, fMRI, DTI, TMS. So we will be discussing each of these in detail. Now, neuroimaging or brain imaging helps in diagnosing any injury or disease in the brain. It could be tumor, it could be cyst, or it could be certain structural deformities. And for that, we use neuroimaging. Okay, so functioning imaging helps in understanding the function, for which we have fMRI, and it also helps in diagnosing disease related to metabolism or brain disorders. So now first we will discuss X-rays. So X-rays are something I feel at one point or another all of us have gotten an X-ray done, be it for getting a root canal for your teeth, be it for a fractured arm or a leg. Okay, if you have not had an X-ray done, you're very lucky. That's really good. But if you have had an X-ray done, you're fairly familiar with the process and what usually happens. So I'll give you an idea. As you can see that is how an X-ray image looks like. And if anyone of you have seen the movie, I think Gajni, Amir Khan's movie, it's a very old movie. If you have seen that movie, they often show X-ray pictures, right, of his brain where some damage has happened and that's why his memory is not working. So that's ideally what X-ray functions like. Okay, so this technique primarily was used to explore the reason behind any neurological deficit that is happening. Okay, so X-ray is a very basic technique which uses photography to understand that what are the underlying conditions if there is something which is happening. So conventional X-ray, often, earlier X-ray which was traditional in nature, it did not help us in understanding the neural activity of a patient, but it only provided us with the structural aspect. Okay, so we know that there are a lot of techniques that are used, but X-ray is something which still sticks by a simple formula of giving you a image of the brain, okay, or whatever part. So it is a type of radiation. Okay, X-rays are a type of radiation called as electromagnetic waves, and these X-ray imaging often creates pictures of the inside of your body. Okay, so whatever is visible right now, we can see the individual's brain, the spine, right, the neck, but we cannot see the structure. We can see the skull and we can see some gray matter, but we cannot see the structure. Same for the teeth. Nothing is clear. So that is what X-ray is. It's about a very surface level assessment that whether something is wrong or not. Okay. Moving ahead, next we have CT scan. So CT scan is a step ahead than X-ray, and this is, I would say, one of the expensive techniques as well. Now, CT scan is a advanced X-ray technique which is used along with computer and helps us create structures of the body and the brain. So this is a huge cylindrical ring. So you might have seen this in movies or shows that a person goes under this machine. So that cylindrical ring is where the person goes, and the X-ray is passed from one side and the X-ray detector is placed on the other side of the patient's head. So what happens is slowly the X-ray and the X-ray detector are rotated and photographs are taken from all the angles, because this gives you a broader and more comprehensive picture. Okay, so a number of scans are taken over here and then they are combined to understand and get a three-dimensional picture of the brain. So neurologists would often use CT scans in case where they can notice where they want to check for blood clots, tumors, sclerosis, or any other internal injury. All right, or if there needs to be a surgery that needs to be carried out. Okay, and that's why we use CT scan. All right. Moving ahead, next we have MRIs. Now, MRIs are one step even ahead. Okay, these are quite fascinating machines and they are used whenever you want to get a detailed picture of the patient's brain. So whenever you are trying to have a detailed picture or you want to investigate further in the brain, this technique is uses. So this particular technique utilizes a very strong magnetic field to gather images of the brain, and it's often that people complain after an MRI they have a headache or they are feeling dizzy. That is because of that. So this technique uses a strong magnetic field and it gathers images of the brain in frontal and horizontal planes. So unlike CT scan, it provides us a more detailed and clear picture of the brain, as I said earlier, and it helps us in knowing the blood oxygen flow to different parts of the brain, as well as which parts the flow is active, which is inactive. The resolution of image is also HD and they're recorded in real time. Moreover, in this particular aspect, you don't inject patient with anything, but in MRI, in CT scan, you might give individual some radioactive substance to ensure the X-ray is being done. But in MRI, that does not happen. Okay. Moving ahead, next we have PET. Now, PET is again something which helps us in detecting the changes in the metabolic activity of the brain. So as you can see in the image, there are different aspects which are red, green, yellow. Okay, so these and help us in understanding when the brain is at rest, when brain is, so let's say, as you can see in different images, when the brain is at rest, there are no changes in the metabolic activity. But let's say if a person is walking, you can see on the left side, the parts are red a little. Then you can see when they are seeing at the back of the brain, the part is red, right? So similarly, this allows us in understanding the metabolic activity of the brain. So it will not provide you with any structural information about the brain, but it does provide you an image of brain activity, which is a functional brain image. Okay. But this is an invasive method where you would be given the injection of a 2-DG, which we spoke about earlier, that 2-deoxyglucose technique. And whenever this is observed by the patient, then we do the check. Okay, so pets can use radioactive chemicals with a short shelf life. So they would often dissolve after a point of time. And this is a very expensive technique. And it's only available most of the times in research hospitals. Okay, so but through this, you can know the metabolic activity on in the real time. Okay. Moving ahead, next we have fMRI. So fMRI is relatively less expensive, and this also tells you about the functional aspect of the brain, not metabolic activities, but it does tap into that area. So functional MRI is something that is less expensive, less risky, and focuses more on understanding the changes in blood flow or hemodynamic response. Okay, so fMRI primarily measures the hemodynamic response, and it is one of the most popular methods whenever you are trying to understand or study cognitive sciences, cognitive neuroscience, or you are trying to diagnose or form a diagnosis for someone who's suffering from cognitive neurodegenerative disorders or cognitive ailments. Okay. Next is DTI. So when we talk about DTI, this is a method which identifies those pathways along with which water diffuses. So this is called a diffusion tensor imaging. Now, you might be wondering, this is a weird technique, right? So what happens here is that we in our body, water diffuses in the tracts, okay, which is called as bundles of axon. So diffusion tensor imaging gives images of these tracts where water is being diffused. So this helps in determining the location and orientation of neuronal axons in the brain. Okay, and this method gives an idea of how different areas of brain are connected to each other, as well as this is also one of the major advantages of this technique in comparison to other is that's not that non-invasive. Plus, it tells us in understanding, see, none of the other techniques allows us to understand how the brain is connected, but DTI is the only technique which helps us in understanding how brain is connected. Okay. Next is your transcranial magnetic stimulation or TMS. So this is a technique which turns off a part of your cortex by creating a magnetic field. Cortex is something which is present in the front of your brain. This is also something that we will be discussing ahead in our future units. So this, what happens is that in TMS, your cortex would be temporarily deactivated, in a sense how you deactivate your Instagram sometimes when you are upset. So that is something which is done. So it temporarily inactivates the neurons below the magnet, and researchers often use TMS to as a mapping technique. So let's say when you place a TMS coil on the motor cortex and stimulate different regions along this cortical side, wherever the motor cortex lies. So you try to see the muscle movement and twitches that would happen in the body or would not happen on the contralateral side of the body. Okay. So through this, we can understand how different body parts map onto different parts of the cortex, and also how much cortex is devoted to any particular body part. So this is primarily something which allows us to understand the functions of cortex, as well as which aspect of cortex is dedicated to which body of yours. So let's say if my subcortical region or my muscle movements all come from my motor cortex, my language comes from my linguistic cortex, right? This is something that we've discussed just for an example. That's what we try to understand using TMS. However, one of the disadvantages of TMS is that it is not very effective in reaching the subcortical structures that are below the cortex or that are in diffusion of cortex. Okay, that is something that we will be learning ahead. All right. So this is what we talk about in neuroimaging. Then comes our neuropsychological assessments. So when we talk about neuropsychological assessments, these are one of the ways through which you can assess an individual's behavior and their cognitive functions. So neuropsychological tests are often applied to make an assessment of different cognitive functions, and this includes different comprehensive examination of your behavioral, motor, psychological, linguistic, neurological assessment of the patient. Okay, so you don't just take one part, but you try to take different aspects of the cognitive function and then try to understand it. All right, so few of these assessments include your Wechsler's intelligence scale. So this is something that was published in 1939 by David Wechsler, and it measures the intellectual performance of adults. Before we get more into neuropsychological assessment, I want you to do one small task. Try to find out all the neuropsychological assessments that have been developed in India. Okay, one I will give you, Nimhan's battery of neuropsychological assessment. So Nimhan's battery is a test which is developed by Nimhan's National Institute of Mental Health and Sciences, right, which is in Bangalore. Do check it out. It is great for budding psychologists, and if you are someone who wants to get into brain or clinical psychology, it's a great institution for you all. And check out Nimhan's battery of neuropsychological assessment. Okay, that's something that will really help you in understanding the Indian aspect of these assessments. All right, so coming back to the Wechsler test, so this was an intelligence test that was developed. Currently we are running the WAIS 4 version, and there have been revisions over the year. And Wechsler also has a specific test for children which is known as WISC, and there's also a test which is called as the Wechsler preschool and primarily scale of intelligence. So this is designed for children between the ages of 4 to 6.5 years. We also have batteries such as Wechsler memory scale, which only assesses the memory, different aspects of memory. Okay. Next, we have the Halsted-Ration neuropsychological test battery. So when we talk about the HRNT, over here, this battery consists of a set of neuropsychological measures. What is the difference between test and a battery? Can you guess? So battery is something which consists of multiple tests. Okay, so there will be a lot of tests in a battery, but test is a single assessment. Okay, but a battery consists of multiple tests. Like there will be tests for memory, there will be tests for motor, there will be tests for cognition. All right, so that is what a battery is. Okay, so most of the neuropsychological assessments are also batteries, because neuropsychology in itself is a multifactorial unit. It's not a single unit, and hence you need to assess all the factors. Okay, so the battery is designed to assess all major cognitive, sensory, expressive, and motor functions, and the test battery is found to be reliable both for young and old individuals. This was developed by Ration and Halsted. Okay, then we have the Lurie, Nebraska neuropsychological battery. So this is a standardized battery developed in 1986, and as you can see that this particular battery is used, again a battery, which means that it has multiple tests, and this neuropsychological test is found useful in diagnosing and treatment of brain damage or dysfunction. So that is where we primarily use this particular battery. Then we have the Ray Ostrich complex figure test. So this test was designed by Ray in 1941, and this is used to assess your visual perception, visual memory in brain damaged individuals. So if you have brain damage or due to any reason, brain trauma, brain injury, this test is often used, and it is one of the most used tools to assess constructional abilities and non-verbal memory tests. Okay, so this can be used from individuals ranging from 4 to 15 years and 16 to 60 years of age, because this is what the norms are available. So for children, we go from 4 to 15, whereas for adults, we go from 16 to 60. Okay. Moving ahead next, we have the Dellis-Kaplan Executive Function System test. So when we talk about executive function system, it includes all the major systems of our brain, which is thinking, inhibition, problem solving, planning, impulse control, abstract thinking, creativity. All these concepts come under this particular test. Okay, so over here, this is often administered individually or on a group, and it is a standardized measure to assess these executive functions on children to adults 8 to 89 years of age. You might be thinking, why such a broad age range? Because executive functions deteriorate with age. Okay, so there will be a peak of these executive functions, but after a point of time, they start to decline with cognitive decline. Okay, next we have a digit span test. So when we talk about digit span test, as I said, this was a part. See, WAIS has a lot of tests in it, and this was a part of the WAIS. However, the test aims to measure an individual's working memory and assess any impairment of the working memory. Okay, so over here, this digit span test often is included in your WMS, Wechsler's memory scale, as well as your Nimitz battery. All right. So this is something which is used. And then we have the Wisconsin card sorting test. So when we talk about the Wisconsin card sorting test, this is a neuropsychological test of set shifting. So what is set shifting? That, you get into a certain set of thought that you're unable to shift, or you're unable to change due to difficulties or dysfunction in the frontal lobe. So this is often used to assess frontal lobe dysfunction in patients of brain trauma, brain injury, schizophrenia, or any other mental illness. So this over here, what happens is the participant has to sort 64 cards to match either with color, form, or number of figures, more like categorization. And participants is told the rule that they must refer from the feedback received from the person who is administering the test. So over here, they're often what happens is, when you try to categorize certain things, you form a mental set. So to understand this shift in set, this test is administered. Next is your dichotic listening test. So when we talk about the dichotic listening test, it's a non-invasive testing technique which is often utilized. So dichotic means two ears. Okay, remember it like that. So this is a non-invasive test, and in this what happens is that you try to investigate for selective attention. However, later on you try to assess the hemispheric lateralization of language ability. So when we do our unit 4, that is where we will primarily discuss brain lateralization, and that is where this test is majorly utilized. So in a standard dichotic listening task, a participant would be presented with different audios through the earphone. So one would say something else and the other would say something else, and based on the instructions by the experimenter, results are concluded. So this could be that over here random words are playing in your ear, and on the other side you are listening to music. Okay, so they might ask you different questions pertaining to that, and depending on your performance on the questions, the assessment is made in terms of hemispheric lateralization. So this is primarily used when a researcher is trying to understand the hemispheric lateralization of the brain, as well as when they are trying to understand how and which part of brain plays in the language. Okay, so that we'll discuss when we come to our unit 4. You'll find it really interesting. Okay, let's move ahead to the research ethics in biopsychology. So we spoke a lot about research and we spoke about different aspects. Something that I want you to reflect on if and when you decide to be a researcher, because a psychologist always have to be a researcher, you would engage with human participants. At times, you would also engage with animal participants, depending on your area of research and your focus. Something that we have to take into notice and we have to be considerate about is how we treat others. So there are certain rules and regulations that should be followed, and you have to make sure that you are respecting the integrity of these individuals who you are choosing as participants. So when we talk about human participants, whenever you are opting for human participants or you are trying to, include human participants in your study, be it thesis, dissertation, your practicals, make sure that you treat them with integrity, respect, and you don't do any harm to them. You give them the proper informed consent. You inform them that, okay, these are the things that would be involved in the test, or this is what will be happening. Okay, informed consent is really necessary. That is where they declare their participation. All right. Next, always make sure that you care for the participant and you disclose all the information that is necessary. Okay, side effects and benefits of the study should be told to the individuals, and ethical considerations as per the American Psychological Association must be followed in any research which is being done. Those are our basic researcher ethic in terms in the field of biopsychology. And apart from that, certain research ethic rules that you should always abide by is have integrity, respect your participants, follow the rules set by APA to do research which is ethical, make sure of regulations, be just toward your participants, ensure their dignity, confidentiality. Whatever research you are planning to do, make sure that you're competent enough. If not, always seek help, always go to your supervisors and get in check with them. Have moral principles, ethical principles, have your own value system which guides you towards the right and wrong. Okay, make sure you maintain the privacy of the participants, you're honest with them, you're making sure that benefits is maintained, that they are having more benefit rather than harm. Make sure you're giving them the autonomy to opt out of the study, and you are not doing any harm to them, or no major harm would be caused to them by being in the study. You might be wondering, but how would I ensure all this when I use animal participants? Because that's what happens in biopsychology. It's okay. Because in biopsychology, specifically, if you need animal participants, there is always a regulatory body that will be making sure how you are utilizing these animal participants, as well as whether they are being subjected to undue pain or not, or whether your research really needs animal participants. So you'll have to go through a lot of hurdles to come to that aspect. Okay, so that will be all for our today's unit. And with this, we have wrapped up our unit 1, where we discussed about the different elements. And in our today's unit, we spoke about various brain research techniques and research ethics in biopsychology that I hope you all abide by as future researchers and psychologists. Moving ahead to a quick let's discuss round, I want you to answer these questions and write answers from your exam point of view. What is the nature and scope of biopsychology? So we discussed the different connections of biopsychology with neuroscience. Try to connect those, try to write it in that way. Make sure you introduce and define biopsychology and then move forward. Delineate the different type of research methods which are utilized in biopsychology. So explain each one of the method from invasive to non-invasive to neuropsychological assessments. All right. How are neuroimaging techniques different than neuropsychological assessments? So neuroimaging techniques usually require either magnetic activities or electrical activities, whereas neuropsychological assessments often require pen and paper administration or some experimental administration. Do give examples of each and explain the difference in each as and when you move. And what are the research ethics in biopsychology? So as we discussed these different ethics, that is something which you also have to take into consideration when you write your answer. Define what is biopsychology and then further define the ethics that are present in the field. Okay, so with this, we have come to an end of our lesson. Make sure you read lots about this, go through your SLM material and be ready for the next unit. All right, till then, happy learning and take care, everyone.